Assalamu alaikum, dear students. I hope you all are fine with the grace of Allah Almighty. This is APSEC's online support program. The video tutorial is for Biology Class 8, presented by Maliha Naeem Khan from Garrison Boys High School, Lahore. This lecture having the remaining topic of the chapter 4, Electron Microscope and the History of Formulation of the Cell Theory. Resource required are textbook and internet facility. The main objective of the lesson are following. Student will be able to get the knowledge about working and the type of the electron microscope. Student will also able to understand the cell theory formulation and the contributions of different scientists in cell biology. There is a recap of the previous topic that magnification of the light microscope is the 1500x and the resolving power is the 0.2 micrometer. But the drawback of using the light microscope is that it cannot show the details of internal structure of any cell. So this is the start of the main topic, that is the electron microscope. It is also presented as EM. It is the most advanced form of microscope. Object and the lens are placed in a vacuum chamber. Electromagnetic lenses are used in an electron microscope. The resource is the electron beam that pass through the object and then it make the image. Electron microscope has much higher resolving power than light microscope. The magnification power of the electron microscope is 2,50,000. Under special condition, electron microscope have ability to detect individual atom, so cell, organelles and even molecule like DNA and the protein are much larger than single atom. The resolving power of the modern electron microscope is 0.2 nanometer, and the 1 nanometer is equals to 1 by 10,000 millimeters. Biologist uses basically two types of electron microscope, TEM, which is abbreviated as transmission electron microscope, and SEM, which is abbreviated as scanning electron microscope. The basic difference between these two types is in TEM, electrons are transmitted through sample, while in SEM, electrons are reflected from the metal coated surface. TEM is used to study the internal cell structure, while SEM is used to study the surface of the structure. There is a question to assess your knowledge that can electron microscope see living organism? Is it so? Yes, there is a limitation of the electron microscope as well, that electron microscope is not used to study the life processes because the sample must be placed in a vacuum chamber, means all air must be removed. So to study the life processes, we must go for the light microscope.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. 1. All organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day, something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
So this is the main point of the cell theory, that all organisms are made up of one or more cells. Either they are unicellular or multicellular. The second point is the cell is the smallest unit of life. The unicellular amoeba is surrounding an algal cell to get the food and energy, as you can see in our picture. The third and the last main point is all new cells come from pre-existing cell. There is a crossword for you guys. To check your knowledge of history of formulation of cell theory, you have to complete it. Next topic is the subcellular or acellular particle. All the viruses, prions, viroids are considered as the acellular or subcellular particles because they do not have any metabolism inside them. But they have able to increase their own number. So somehow they show the character of the living organisms. Yes, we are facing today a disaster due to a virus. So this is the whole summary of the today topic that electron microscope have high resolving and magnification power than light microscope. Electron microscope is not used to study the life processes. Cell is the basic unit of life. Viruses are not considered as cell, and that's why they are not classified in 5 kingdom classification. This is the assigned task. You have to answer these questions. I'm going to end the lecture by the message of the Fred Rogers that look for helpers. You will always find people who are helping. Thank you so much. Stay safe. Allah Hafiz.